I want to walk a little bit through our mode of action and discovery work to get you a little more comfortable with how SOURCE is working. The first way we look to confirm our mode of action of SOURCE increasing nitrogen activity is by measuring the expression of gene activity of 3 known nitrogenase subunits. Nitrogenase is a nitrogen-fixing enzyme converting nitrogen gas to ammonia. And generally, it can be said that the more gene expression will lead to more enzyme produced, which is a potential for nitrogen fixation. So in the presence of SOURCE, you see that gene activity of those 3 known subunits increasing, indicating that the potential for nitrogen fixation is increased in the presence of SOURCE, indicating SOURCE switches on these microbial genes for nitrogen fixation. What you're looking at here is the indicator of nitrogen fixation in the presence of SOURCE and in a control treatment. The white bar again is the control, and the yellow bars are the treatment of SOURCE at increasing concentrations as you go to the right on the chart. So at increasing concentrations, you see an increasing activity of nitrogen fixation. This is the gold standard lab assay used to measure nitrogen fixation, and so it's one of the key indicators that we use in our mode of action classification. And finally, what we'll look at here are soil microbes that were cultured in nitrogen-free conditions, treated in a control, and then treated with SOURCE. So the composition analysis shows that in the control treatment, 2 known nitrogen-fixing bacterial species, Azotobacter and Pseudomonas, were detected, but not abundant. In the presence of SOURCE, these 2 known nitrogen-fixing bacterial species are abundant and detectable in nearly 50% of the composition in this sample. So this gives us a great amount of assurance and confidence that SOURCE is indeed enriching the nitrogen-fixing bacteria in the soil. Switching over to the phosphate side, we know that SOURCE is activating phosphate solubilization, unlocking that P that was made unavailable from previous application. And the way that we measure that in the lab is in the form of plant-available phosphate in a control and in a treated environment, treated with SOURCE. So you see here in the presence of SOURCE, the available phosphate is about 1.7x times that of the uncontrolled bacterial sample, giving us a very good indicator that increased plant-available phosphate means increase in the phosphate solubilization activity. And finally, I want to run you through some of our measures in the lab through multispectral imagery to show the impact that SOURCE has on plant health and plant stress. So we look at things like digital biomass, greenness, NDVI, and then the converse of NDVI, which is plant stress. So you'll see the indicators in the yellow bars, the treatment of SOURCE, over a 1.5x average increase over control in the presence of SOURCE. You see that matching up with the photos on the right, the control plot being on the left side of that photo spectrum and the treated SOURCE plot being on the right. So you see a greener, healthier plant, and then you see an expanded root architecture in the treatment of SOURCE.